Bring around the microphones for the questions.
Yeah.
Hello, everyone. Welcome back. Here we are again. As you may have learned, Finn has done some amazing respiration analyses. And here we are learning more about how we can also implement them with this special session called Breathing Together in Music, a RESPY workshop by Finn Upham.
Thank you. Everything should be working in Audible, so hopefully things will be good. I've added myself to the corner up here if you have trouble seeing me over all of the screens. But yeah, it should be pretty clear what's going on. So through this, I'm going to talk about some basics about human respiration, and then introduce the RESPY Python library and talk about how this system particularly treats respiration phase. And then we can kind of negotiate how much depth I go into respiration and music listening, phase alignment and music listening, respiration music playing, and phase alignment and performance. But I want to get back to this question at the very end, which seems very appropriate for this workshop, this larger workshop, which is this question about whether the system, the respiratory system, actually entrains or not. So we'll get there. Based off of what I show, you might guess what my answer is. But I also respect that we've got many different definitions of entrainment out there. So we'll try to get into the specifics of what and why that is working how here. So that seems like, well, we'll just have people wondering in. Okay, so first up, human respiration. One of the reasons why not a lot of people look at respiration is because it gets affected by kind of everything. This is a diagram that is trying to attempt to integrate all the different systems that can influence how we breathe and when we breathe. But there's even actually things missing here. So it mentions locomotion. When you're moving, that can influence how you breathe. Obviously, if you are doing a lot of work, and if your circulatory system is really active, you're going to also need a lot more ventilation. So that's going to be really prominent. But we also have things like speech and emotional expression that become very strong influences on the how and the when of the respiratory system. And what's missing here is that we also have the auditory system that influences when and how we breathe, because breathing is noisy, and that means that it can mask noises in our environment. And so there is also some influence there to stop us from breathing when we really need to hear. And so you just have all of these influences that are negotiating the how and the what of our respiration, which means that when we measure respiration, sometimes it's hard to figure out why breathing looks the way that it does. But it isn't a lost cause. It's just complicated, and we need good environments to control how we assess it. So I'm getting these notifications about people in the waiting room, hopefully. So we can turn off the waiting room. That should snap it off. So you go to security. Oh wait, because you're not a co-host. Oh, I am co-host now.
Okay, so now you can disable the waiting room, which is here. Click that. Oh, click it, and then enabled, and then unclick. Admit. And then hopefully it stops doing that. And I'm also gonna try getting my
okay. All right. Sorry about that. Zoom is complicated. Okay, so but the respiratory system is more complicated. I'm sure we can win at Zoom. I'm not sure if we're going to win at this. But one of the things that comes along with the respiratory system being so involved with so many systems is that it's a very flexible system. And so there are many different ways to breathe to cover your bases, to make sure you are doing enough ventilation to maintain homeostasis. So disruptions are actually very highly tolerable, except in those situations where you're like really desperate for breath, in which case, you know, that becomes more urgent. But most of the time, we've got a lot of flexibility here in the system, which means it can be a very rich indication of the stuff that is happening and the stuff that people are doing. When we're looking at respiration, or the respiratory traces that I focus on are chest stretch measurements, and they are a proxy for lung volume. It's not a very good measure of lung volume. If you really want to have a good estimate of lung volume, you know, you put somebody in a mask and you track the airflow going in and out of their body. But that's very distracting and disruptive. And so a single elastic band around the chest at least gives you a mostly monotonic kind of correlate to the ventilation, to more air, less air. Now, this is a description of sort of what is happening in a respiratory, for a couple of different respiratory actions here. So first off, we have a normal breath. And we have, this is tracing the volume of air in the lungs. And so it's kind of in this sort of middle range. And then as you breathe in, you expand, you get more air, and then you crest and you exhale. And then if you're engaged in something like singing, usually people breathe in a lot more and very fast, it's faster. And then the descent is very controlled and will go past this threshold of stability. So every breath that we take, we move away from a point of stability and then we return to it on exhalation. And if we're not doing something strenuous, that exhalation actually doesn't involve any muscles. It's just a relaxation of the system. So generally speaking, the kind of breathing we usually do, the kind of breathing that you're probably doing right now is an active contraction of muscles in the diaphragm and the external costals to increase your chest cavity that allows for air to come into the system. And then those muscles relax and the elastic tissue in your lungs will pull the lungs back down to your kind of resting state. And so when we're looking at respiratory timing in particular, it's really helpful to understand that there's one part of it that is controlled by muscles and the rest of it just happens. Now, of course, when somebody is singing, we have a much more complicated constellation of muscular controls that is counterbalancing. Some of the inspiration muscles continue on. The expiration muscles allow for this very controlled descent and flow of air during singing, obviously during wind playing and a bunch of other things during talking, mostly. But this is a very different configuration of muscle controls and of an action program than what we see when people are just kind of sitting there and listening to whatever's going on. Now, when we look at single chest stretch sensor measurements here, we can clearly distinguish between these two types of breathing, even from this sort of very basic, like stretch in and out kind of measurement. So above here, we have a kind of normal breathing pattern from one person listening to some string quartet music. And you can see you've had this clear inhalation, that's the active part of it. And then you kind of have this sort of wobbly collapse back to stability. And then we have these sort of gaps here between breath to breath. And the gaps are kind of different sizes, but the inhalation and exhalation are not perfectly symmetrical, but they're usually kind of a little bit symmetrical, with exhalation being a little bit gentler. Whereas when we look at somebody singing, this is somebody who I didn't ask to sing, they just sang along to what they were listening to. So this is entirely spontaneous. I don't know how well she was singing, but she sang professionally, so it's probably legit. Anyway, so we have these strong, deeper inhalations and then these controlled, smoothed exhalations that continue on what would happen otherwise. And that's reflecting that sort of different kind of pattern of control that's related to the action of singing while still moving air in and out of the lungs. All right. So when we're looking at respiration, this is the kind of information that we want to have. We want to have information about when a breath starts. We want to have an idea of what kind of breathing they're performing. And maybe it's important to know whether, for example, air is actually moving through the body and through the mouth, which is very close to the ears, or whether it is kind of stable and not changing and airflow is low. So that's why I developed RESPY. So this is a Python library that initially was developed in MATLAB, and all of my PhD research was in MATLAB, and I've ported everything into Python, and I've been working on it for a few years, which means it's not guaranteed to be stable, but it's like pretty good at what it does. So I'm happy to share it now. But just a warning, this is a project that is by me, and I am not trained as a software engineer. So I'll maintain it as best I can, but please be kind with your comments and bug reports if it comes to that. I'm happy to improve it, but there's also limits to its structure. So anyway, but the point of it is firstly to be able to clearly detect breaths. So as opposed to, for example, trying to get a frequency estimate by taking an FFT of a smooth signal, which is another way of capturing information about respiration. This is actually going to try to find breaths with a high degree of precision, and then identify phase components and the timing of it to be able to look at that in relationship to what is going on, and then be able to give important features about the respiratory cycle, and then categorize, or at least begin the process of categorizing specific respiratory actions. So we can say somebody is singing, or someone is breathing in the way that they would potentially be singing, even if we can't be sure exactly what they've done. So first, let's just go over here. So RESPY, this is, I don't know, how many people here actually program in Python? Okay, all right. So you guys could all go in there and do pip install RESPY, and there you'd have it because it's up there as of 2 days ago or yesterday. Well, I don't know what the date is. Anyway, very, very fresh. I had it on the test site for a long time. So it's just fresh on Pipey. It's not fresh, period. But to get into how this thing works, I'm going to be sharing things from this demo repo that I've also shared, and it's been posted actually in the program. So if you want to just clone that, that'll give you the same stuff that I'm running here, and I'll be running some code live. So hopefully it'll work just as well for you as it worked for me, as long as you can work out the dependencies, because I didn't quite manage to control that properly. So in here we have the RESPY introduction to the RESPY functionality. And by the way, since you're all here, I can let you in on a special little secret that's not really secret because it's in the code. Besides the code that is actually currently running through the pip install, you can access all of these same functions within this particular sub sort of file here. So I can explain things here, but you can also see fairly well documented descriptions of all the different functions in this other file that is exactly the same as what has been submitted through, and that can be downloaded as a proper module in PyP. So what does this do? First off, let's just get these things sorted. Within RESPY, I also kind of snuck in a second code library that I haven't really released properly. And that is the activity analysis library. That's a statistics library to look at the coincidence of events across collections of concurrent time series measurements. So for example, if I want to look at the sort of the significance of the alignment of inspiration onsets, I can do that with the activity analysis toolbox. So that's why I bundled it together, because we'll get to that. But that's there for free inside PyP if you install it in the normal way. So to just show how this works, I'm going to load some data. This is 2 recordings from one person as they were standing in the mocap lab, listening to music play on speakers and then silence, and speakers and silence, and then headphones and then silence, headphones and silence, or maybe it was, yeah. This is measurements from a chest stretch sensor that's measuring things only at 10 Hertz, not super high, but it's enough for us to work with here. So if I pull out one of those columns, I haven't defined sample duration. That would be good to do. Let me just do that. We'll say it's 120. Perfect. Oh, it's in the next one. Right. Okay. So here, this is an example of the chest stretch measurements. Now, these measurements actually have been normalized. So the first function that's worth pointing out is that there is a function here to, by default, normalize respiration. Chest stretch measurements are by default kind of unitless. There's all kinds of different ways of measuring it, and it's kind of fuzzy. So what this does is it looks at the amount of sort of when it changes, and it normalizes based off of the difference of the data, specifically the differences on inhalations, because that's kind of the most stable, reliable part. And it's not going to perfectly normalize things, but it will scale the signals to be kind of the same, even if one person has a slightly more loose chest stretch measure than somebody else. This is very helpful for that. And here we can see the respiratory sequence. We've got some normal breaths. We've got these things. They're called augmented breaths. They're something that we do kind of all the time. It's just a sort of a double-sized inhalation that needs to, it's good for your respiratory health. I don't need to get into it. Anyway, it's important. It's good that it's happening. And there's the kind of normal sort of noisy deviations. And then we have slower breathing and faster breathing and so on. So now we can look at features of these individual breaths as plucked out by the system. So the first thing to notice that the inspiration extraction function will pull out the onsets of inspirations. And this is the onset of inspirations after filtering. So there's a little bit of a shift here that happens when you do filtering. It's a low pass filter. You can control the kind of filtering. Here we're using a 1 Hertz threshold. If you can raise it and lower it and avoid some of that interference. But anyway, this is what we're using in this case. And this gives the precise timings of the onsets, so the minima and maxima, but it's using a special heuristic that I developed after looking at a bunch of different ways in which people try to pull out inhalations. This is the one that performed the most reliably when I was looking at measurements of people just sitting there. So it's not perfect, but it is pretty good at identifying actual onsets of inspirations rather than the extra squiggle that comes from just using a basic minima, maxima kind of tracking thing. And it gives you all the timing of those. And it also gives you the timing of the corresponding expirations because that's part of the algorithm. So now we also can pull out a bunch of statistics about each of those breaths. So breath features, pretty self-explanatory. Here we have obviously the inspiration expiration timing. We have depth after normalization. So this is again a relative value that has something to do with lung volume, but we'll just kind of leave it at that. There's also here 3 timing intervals. So we have inspiration time, the full period of the respiratory cycle, and then expiration time, where expiration time is just the full period minus inspiration. And there's a couple things to note here. One is that if you look at this row, this is in seconds, and the inspiration times are pretty stable for this measurement. There's some extremes, but a lot of the time, most of the time, it's around 1 second. And that's kind of how it is. Inspirations, unless you're breathing for action, it's about a second. It's maybe mostly a little bit less than a second, but it's just kind of around there. And that's one of the most stable components of the respiratory cycle without action. But if you see that sort of there's some variability, but not a lot, there's actually a lot more variability when we look at the expiration time. You know, sometimes this is like 2.5 seconds, up to 4 seconds. There's a kind of a breath by breath, a fair bit of variability. And one thing I want to point out here is that if we were trying to match music to the natural respiratory cycle of a person who is just sitting there not doing anything, we would not be able to maintain a good metrical hearing of that respiratory cycle because there's just a lot of variation from breath to breath. It's more than would be comfortable. You'd have to do a weird kind of adjustments to match the slightly longer and the slightly smaller. And that variability makes the kind of, when we think about like how respiration behaves against music, one of the things that's a difficult reality is that it is less regular than music needs to be. A lot of musical behaviors need to be very precise. Respiration, in a lot of cases, doesn't care that much. So it has other priorities. Okay, other features here is the inspiration-expiration ratio. That's a very common statistic in respiration research. It's one of the fast ways to be like, oh no, that's speech, because the inspiration-expiration ratio is actually really high. Or is it low? Anyway, whatever, the statistic is the right one. And it's a good check. It's kind of the rapid check of like what kind of breathing is happening, and it's because that ratio, it's the expiration that's the most flexible here, indicating that stuff. All right, those are those features. I can also extract those features in a way that's sort of continuous, so we kind of interpolate it. It's a step function interpolation, so we can actually see continuously. Here we go, the same sort of, this is a 60-second excerpt, and we see breath by breath the timing of the inspiration, the expiration, and the period. And we see, for example, just between them, there's some differences here, like the inspiration contour goes down before the expiration contour goes down, and all kinds of things. So there's a little bit of separation here between these signals. And which again is sort of like, well, you know, this is while somebody was listening to, I think it was just a metronome, and the metronome didn't really, it wasn't something that was drawing their respiratory system into alignment. Okay, so this is a kind of a normal way of looking at respiration, but because respiration has all of these weird things about having this sort of variable length expiration intervals and these quiet periods, and we know that mechanically these quiet intervals after your expiration, there isn't anything that is pushing or trying to determine the timing of the next inspiration after you breathe out. So you have a kind of like a general need to keep breathing, but it's not like when you breathe out, there's an automatic trigger to start breathing in again, as you would expect from a normal mechanical oscillator. And so there could be useful information, particularly since we know that inspirations get suppressed sometimes for auditory attention and whatever. There could be actually useful things, and particularly focusing on those low flow intervals, because that's where it's kind of the most flexible. So instead of doing this two-phase thing, what I've chosen to do to study music and respiration is to actually use 3 phases. And I'm not making up the 3 phases. This is actually how biologists look at respiration in other animals. Actually, when you look at like frogs and stuff like that, they usually talk about like 4 respiratory phases, because you have the inspiration and then you have the post-inspiration pause where they're just like holding breath because they're swimming or whatever. And then there's the expiration and then the post-expiration pause. So even though normally for humans we talk about 2 phases, there's lots of conditions and lots of respiratory systems that actually require more segments. Now, for humans, a post-inspiration pause can happen, but I'm not studying divers, so it's fine just to have 3. So there's the inspiration, the expiration, and the post-expiration pause. The other thing that ends up being quite useful to look at is, so one of the things that distinguishes these is not just how regular or the duration of them, it's also the physical sensory consequences of these phases. So I've mentioned auditory masking as being an important thing, but when your breathing is moving in and out of the body, there's also kinds of sensory things that are part of it as well. So there might be situations where we want to have that feeling of air. It might be of value, it might be of use. In which case, maybe the high flow ends up being useful to distinguish from low flow intervals. It's also very difficult to figure out when exactly the post-expiration pause starts. It's a very ambiguous thing. And so instead of having a like firm transition on that, the way that I look at these components is I extract the onset of inspiration. Here, actually, I'll just do this. Boop. So I track the onset of inspiration, because that's the thing that we can find in the signal clearly. The interval of high inspiration flow, which is actually usually the full inspiration interval because it's a pretty steady drawing in of air, usually. The onset of expiration, because that's when the system relaxes and there's a clear peak there that comes out of the measured signal. And then this interval of high flow expiration that kind of peters out. And then another threshold that defines the sort of like, well, it's probably low flow. It's probably low flow is very hard to get very firm lines on, in part because we're using chest stretch sensors. It's kind of ambiguous, and that's also where there's the most noise in the sensors, because that's when the sensor is relaxed. So these 3 components end up being kind of essential for pulling that out. Let's see if we can plot it. Oop. There we go. Yeah. So using, this pulled out respiratory phase information, and then this is plotting the inspiration onsets, expiration onsets, and also these flow components so that we can see this is the initial measurement. And then these are where I've identified these phase things. And you can see, I'm not sure if this is, actually I haven't color checked this, but there's a little bit of green. Here, I can actually zoom in. Woo, zoom on zoom. Okay. There's these little green intervals, and those are the post-expiration pauses. Those are those cases where sometimes it's long, sometimes it's really short, sometimes it's barely there at all. And it's this flexible period or this section of the respiratory cycle, while the inspirations are pretty stable and the expirations are just kind of a natural consequence of the inspirations. All right. Now, with this way of breaking down respiration, we can then look at, well, so does this mean anything for music? This is the basis of how we treat the signal. So let's get back to musical conditions. So I'm going to actually, before I get to the concurrent respiration stuff, I'm going to flip to respiration and music listening. So in the process of doing studies like this is actually a collaboration with Alexander and Agnieszka and Victor, who were here a couple of years ago. They collected data of people standing and listening to music and then listening to silence, and they were listening over headphones or speakers, and that condition didn't really matter here. What we have is just this contrasting state of there is sound in their environment that's musical, or there's just silence while they're standing there. And one of the things that came out of looking at respiration in this segmented way is a recognition that there's this clear, apparent distinction between the respiratory phase, the respiratory structure in silence versus sound. And one of the ways in which that comes through is in the respiratory period. There's a significant difference here in the inspiration period. But another way in which that comes through is actually in the sequential difference. So not only are respiratory periods generally shorter when people are listening attentively to things that are happening around them, but they also tend to be more consistent. There's less variability. Silence results in a much more variable respiratory behavior, probably because people are just in their own heads. And so the respiration has to do with what they're thinking about. Whereas in the context of sound, in the context of music, maybe in the context of, I haven't checked reading. I think this comes through in like movies and stuff like that too. There is a shift of respiratory behavior that seems to be a little bit more consistent and a little bit shorter and soft as well. This isn't a fast breath. It's a kind of clean, subtle way to keep breathing without interfering with your experience or other people's. And this is a reliable category that comes through when we're comparing silence and sound conditions. So we know that we can look at it at the feature level of how that happens, but we can also see how that gets negotiated. So this is actually trying to then focus in on when there is the onset of a sound, how do we get a shorter period? Where is that shift happening? When does that happen in respiration? And one of the things that we can see here is that the change to a shorter interval is noticeable both in the inspiration and in the expiration. And so, these are sort of whether somebody was in the process of inhaling or exhaling as the music started, either way, they kind of got tighter in their respiratory cycle very, very quickly. So within a respiratory cycle, this isn't a gradual shift to normalcy. This is a very prompt shift in respiratory behavior in response to there being something to pay attention to in their environment. And what is a little bit funny is that when we look at metronome offsets, the inspiration doesn't seem to change a lot, but the expiration does. So when the music stops, the expirations very rapidly relax and they go back to this longer sort of cycle, because the difference between quiet breathing and this sort of not quiet, however you breathe in silence, is mostly in the expiration phase. It's that kind of stability there. So this is coming through quite clearly that this is a rapid change of respiratory behavior in response to what's in the environment. Okay, this is a toy example, but this does suggest that like we change and adapt our respiratory behavior very rapidly. So let's get back to this question of respiratory alignment to music. So here I'm going to pull out an example that comes from a repeated response experiment I did quite a while ago. This is one person listening to the same piece of music many times over several weeks, and we've normalized those signals. Have we normalized those signals yet? Yes, we've normalized those signals already. And so it's not perfectly aligned, but they're kind of in a similar scale. And if you look at these respiratory traces, most of the time it looks kind of like normal. I mean, it's hard to see here. And there's a couple of times where there's this like long pauses as they just don't breathe for a while, which people do actually. Don't get alarmed if you're looking at respiratory measurements and people just kind of don't breathe a lot for 20 seconds. We can take it. It's fine. It's not actually it's not like their heart doing that. That's a whole different story. Respiration is okay. Other systems, maybe not. Yeah, so there's this kind of shifting of the respiratory behavior is mostly like this quiet breathing. It's not super stable, but it's kind of stable. And this is the noise of respiration sort of before the onset of sound. And then this is when sound starts and it gets, more regular. And then the sound ends and you get weirder breaths again. To look at this maybe a little bit more clearly, we can, oh no, now we're doing the normalization. Okay, all right. Well, I'm glad I included that cell. This is the same information presented in difference data. So now I've taken each of those time series and taken the difference of them and then plotted them one on top of the other in alignment against the stimulus. And I think the stimulus isn't here, but what you can see is that there tends to be this sort of like, when the music starts, there's not actually a lot of breathing, which is kind of weird. And then we get to these intervals where it's not like this person always breathed exactly the same way every time, but we have these patches where there tends to be several breathings in a row or across these, where inspirations seem to be kind of at the same time in the music. And that is, to be honest, actually quite surprising. When I started looking at respiration stuff, I did not expect to see this kind of consistency of breathing at a particular time, because this is someone who isn't singing. They're just listening. There's nothing demanding them to breathe differently. But because the respiratory system is so flexible, it doesn't cost them anything to shift their breathing a little bit forward or a little bit back. So that's something that this person seems to be doing. I've been kind of running along here. Any questions so far? No, we're good. Okay. Right. Cool. So then the question is there something, is this just a kind of coincidental alignment, or is this a real alignment? And so to show just quickly how that sort of question gets answered, we can look at the same signal. This is a second document within the toolbox. This is the RESPY phase alignment demo. And we'll just have this one start, get that same signal calculated. And then I will get phase components pulled out here. Right, yeah, good, okay. So then this is actually going to assess whether or not these phase components are actually aligned or not. I mean, when we look at the respiratory signal, there's obviously some kind of alignment that is happening, right? We could use all kinds of statistical tools to tell that, yeah, actually there's more alignment in these oscillatory signals than you would expect normally. But because we know that the respiratory phases all have different meanings, right? Some of them are noisy, some of them are quiet, some of them are driven by muscle contractions, some of them have nothing in them in particular. It's just kind of silence. And so we have to treat each of those differently to figure out whether they're aligned or not. So this is actually going to take a little while, and it's going to evaluate for each of the 5 components the quality of alignment, count the alignment, and then give an estimate as to sort of like how weird is it that they actually do align to this degree over repeated listenings. And oh, scale high not defined. Yep, do 7 and 3. I forgot that I deleted that cell. This is live coding. Sorry about that. Was not expecting to do this. It actually has a default. I could just erase these terms and it would be fine. But anyway, there we go. Okay, this is going to take a couple minutes because what this is doing is it's a Monte Carlo technique that looks at alternative alignments of these signals to see whether they kind of line up the same way by chance as they do in this sort of original alignment. So we're just going to let this, well, there's the preparation, and then this will run it properly. And we'll come back to this to see the live results. We have a chat. Oh, you chaired that. Okay. Cool. Thank you. Can I hide that? Yes. Good. Okay. So now let's talk about phase alignment. So what's happening is it's doing this sort of the activity analysis standard shuffling thing to figure out alignment of these different phases. There's counting and there's alternative alignments. And you can read about that in the paper that actually explains it properly. But for now, what the important thing is that there's a bunch of parameters that this library tends to use for how to evaluate coincidence within each of these phases. And those parameters were selected through a process of figuring out what actually gave clear results of yes, there's coordination, or no, there's not, based off of a clear alternative. So I'm not just like putting in any numbers. This is actually based off of what seems to capture information about these phases when we're looking at this type of signal. Yeah. So does alignment happen? All right. So it seems like it does. This is actually, it doesn't always. This is an example of one person who has a lot of respiratory phase alignment over repeated listenings, and this is someone who doesn't have a lot of respiratory phase alignment. It's different pieces, but if I were to use the same pieces, this first person basically doesn't ever do respiratory phase alignment when listening to music. So that's just kind of one of those things, is that this is a behavior that seems to be related, that seems to be, you know, distinct for specific people, but maybe those people aren't so rare. Let's see. Okay, it's still going. It's got 5 phases. We should just let it.
What kind of people tend to align, or is it that?
Yeah, so I mean, I haven't had the chance to do big scale population analyses of this, but I can say from case studies that it seems like people that normally use breathing in the context of music for music generation show an inclination to breathe like other people that are breathing for music generation. So if you're a singer and you're listening to singers, you probably won't notice, but you may very well be shifting the timing of when you breathe in to be where you would need to if you were singing too. And the same goes for wind players. So thus far anyway. And it's not to say that nobody's a deviant from this, but that it seems to be the pattern, and that would make sense. This is an aspect of the musical signal that people can be sensitive to. And even though everybody breathes, not everybody has the experience of breathing with other people singing. And so with that context comes a facility and a sensitivity to this aspect of the musical signal that can nudge your respiratory system. It doesn't have to, but it can. Yeah. So, yeah, so this is looking at this actually is a good one to show here, because this is a bunch of different people's alignment qualities for the same stimulus. And we have one person who's very aligned, someone who's like, I mean, maybe there's some alignment here in this moment, but it's not very clear. And another person who has a lot of this sort of stripiness, but it's not locked in. It's like, there's like kind of like an opportunistic thing, right? You shift your breathing when it's relevant. And then you get these kinds of patterns that relate to the music. And I'll just play this one because it's a good example of this alignment. So this is the different phase components: onset, high flow of inspiration, expiration, offset. And what these dots are showing is sort of moments where there's a very high consistency across repeated listenings by, I think this is just, this is everybody. So this is aggregating everybody's information. And we can see where their phases tend to be the same.
Je ferai faire un beau bouquet de 3 couleurs de rose. Je l'amarrai avec un fil d'argent. Tous nos amours, ils seront mis dedans.
So this is an example of, it's a very convenient, tidy example. It's not always this clean, but in a situation where there's only voices and where they're all breathing together and where it's very clear when the need is happening next, this is a situation which kind of throws that respiratory system in your face, and you're like, yes, I can follow what's going on and I know what needs to happen and I can do that too. And so some people do that too without having any idea that it's happening. This kind of coordination can also be measured between, not just within people, but also across people. And so we have some instances of audiences that have measurable patterns, and I'm not going to play all of these, but what happens in an audience context, as opposed to, say, listening to a recording, is obviously you have different information about what is happening. And so it could be that what is cueing respiratory alignment may be different in those cases. Because while there is this thing about like hearing somebody breathe and then breathing with them, there's also all these other cases where respiratory phase seems to be weirdly aligned. And so there's other reasons for people to breathe at particular moments with the music. And sometimes it seems to be, because it's so beautiful, but everybody has that, it's so beautiful reaction kind of around at the same time, because music, directs you that way. And then you can capture that across an audience. But it's not the kind of behavior that you can just get in the lab any old day. But it seems to be another one of these moments where respiration is important somehow, is part of the process. We can't say whether it's leading or if it's a consequence of some kind of experience, but it's involved somehow. Okay, so that's respiratory phase alignment to music. Let's see. Oh, look at that. Hey, we've got a graph. Okay. This is the alignment that's being measured on this figure. And we have these like really strikingly sort of, they're not perfectly vertical, but they're pretty strong sort of, yes aligned, no aligned kind of patterns at these moments. And what we see is that if we look, for example, at inspiration, there are these moments where there's a particularly high peak in the inspiration that suggests, wow, actually that's really consistent. And over 12 listenings at this moment, oh, that's like what, 9 of the times that the person listened to this piece of music. So 75% of the time, they did whatever they needed to do to inhale at that particular moment. And that's actually a pretty strong effect on the full scale of like things that people can do in reaction to music. Yeah, we also have a lot of alignment in the post-expiration pause, because that is one of those things that is negotiating where you can do that adjustment to move your inhalation forward or back. So we have post-expiration pause ends up being this sort of really interesting feature for consistency in this type of listening. Yeah. Okay, so that's examples of respiration phase alignment in singing. But what about what happens in playing of music? So yeah, actually we can go back to the notebooks. I have examples of those here too. So respiration in musicians. We're going to pull out a violinist just to show an example. So violinists, obviously, they can make sound without breathing any particular way with it. However, there's also practices of breathing with music playing that a lot of them show. And so their respiratory behavior can be quite variable. So this is plotting an excerpt of this one person's breathing. And what we see here is that the respiratory shapes, those respiratory shapes don't look the same. That's weird. Why is it not looking the same? Is it pulling RESPY 2? Yep, yeah. New phases, onsets. I was very eager to remove, this needs to be up, cells in order to keep this tidy. And I was overzealous. There we go. Those are the right signals. Good. Okay, glad I could fix that. I'll push the change after this. So something that I want to point out here is that this violinist isn't breathing in a way to make sound, but there is a very different shaped breath happening while this person is playing. Sometimes it's big, sometimes it's small. And there's also these intervals of not breathing that seem to be very long. And the not breathing is of a type that seems to be confusing my detection algorithm a little bit. So this little dot is trying to tell me that there's an expiration onset. And like, no, clearly that's just like a continuation of a not breathing interval. So the parameters that I have developed that are part of RESPY are well tuned for respiration when people are just listening to music. But the full expansive arrays in which people can breathe when making music is not super reliable. So it's not to say that you can't use this to pick out phase information, but there's going to be some extra breaths and there's going to be some missing breaths. The onsets that it does find are not bad. But it's an ongoing process to kind of figure out better heuristics that will actually allow us to extend this to wind players and other musicians in a way that is a bit more reliable. But it is important to see that there's a lot of stuff happening here in the respiratory signal. And I'm like fairly certain that this person isn't really concentrated on their breathing in this moment. You know, this is somebody who's playing live on stage. They've got other priorities, but respiration is part of their performing process. And so there is a relationship there to what they're doing. And I'm sure that Laura and I will have a lot of fun trying to figure out more about what that is. Okay, I'm just going to, let's see. 5 minutes left. Okay. All right. Well, then I will share this. This is the same players breathing from multiple performances of the same excerpt. And we can see here that, again, there are breaths that are actually quite well aligned with the music. And then there's a bunch of breaths that are not, even some big ones. This would be a big breath. And the exhalation seems to be aligned, but the inspiration doesn't seem to be. So the kind of relationship that this respiration can have to the music is going to be very complicated, which means we need more people to be studying this. So one of the reasons why we're sharing, well, I'm sharing this library is to encourage people to collect respiration data and then try to do some of this untangling, because it's going to be a long project to figure out how to actually capture this stuff well. Okay. I guess I'm going to show you one more example and then we'll get to that final question. So because it was so much fun the last time, this is an example of this orchestra breathing at the beginning of the Tchaikovsky's Hungarian dances. It's not the full excerpt, it's just a little bit. But it's again showing the kind of diversity of respiratory behavior that is clearly related to what they're doing, but not in a way that is super cohesive. But you'll still see the main patterns. You hear it? It's awkward when you can't see the conductor. You can't fake it. Some of that information is missing. But that just gives an idea of like how rich this signal can be and that we have yet to uncover. Okay, so the last point that I want to focus on, if you ask the right questions, maybe I'll play you the fun example of the orchestra spontaneously singing. The last question I want to get back to is how we think of entrainment and what entrainment is and whether entrainment is really what we should be thinking about when we look at music. I think the violinist's respiratory behavior is a good example of this. That's not an oscillatory system. That is not a simple system that is following an on-off or going around in a continuous phase kind of way. If initially when I started looking at respiration, I tried to use a continuous phase model and representation, and it was so crunchy. It just kept on breaking, and it was a big waste of effort because the math that defines how we understand sort of continuous, smoothly entraining oscillatory systems, there's a lot to the dynamics of those systems that want one cycle to be like the next. The next cycle is trying to kind of continue behaving the same way. And when it changes behavior, it does so gradually and according to rules in a way that you can describe in a systematic way. And when we're looking at respiration in the context of things happening, respiration doesn't really care about the past. It seems pretty clear that respiration is ready to adapt to present needs and maybe anticipated future things. So you can delay inspiration, you can breathe more deeply, you can involve an active expiration if that's what you need. But the respiratory system isn't trying to preserve its past behavior into the future. There's no energy in that system trying to continue on consistency, which means that mathematically, talking about it like an entraining system, like an oscillatory system, is losing a lot of information. You have to kind of filter out all this cool stuff about when phases happen in order to get something like an average respiratory rate. And that is sometimes maybe necessary, sometimes maybe the right information if you're just kind of tracking more ventilation or less ventilation. But for something like music, where we kind of care about when things happen, that's a huge amount of information to lose. And so when in the context of us talking about like what entrainment is, it's very important to recognize that sometimes the information in these systems, while they are happening with the kind of metrical entrainment and all of the entrainment that's part of the process of making music and engaging with music, these signals don't necessarily have that property at the same time. Some of these systems are protected. Some of these systems are separate and behave very differently. And so, in my opinion, you have to have a very good reason and a very controlled environment to treat the respiratory cycle as some kind of continuous phase sort of description. And a lot of the time, particularly in music, that's not the most efficient or useful representation of what's going on. And hopefully with good analysis tools that'll allow us to track phase and actually extract this information more reliably, we can get at the cool parts of breathing and actually learn from it. All right, I think that's all. Thank you.
Really impressive. Thank you so much, Finn. Does anybody here in the audience have questions? Yes, Josh.
Oh, thank you. That was really cool. I was wondering if in your work you have any videos of a orchestra spontaneously synchronizing their breath.
I love planting questions. Thank you.
You're welcome.
This is actually this is one of these ambiguous cases of like, are they actually here, we'll do it full screen. This is worth it. Okay, so and part of the Stavanger Symphony Orchestra, there was an MC that was engaging with the kids, and one of the pieces that was being performed was Festival of Venice, which is a melody that Norwegian kids apparently know already as like a common folk tune. And so the MC was getting the kids to sing along with that. And then, well, we'll see what happens. So.
Trekanter har, og har den ei, så den ei. Bli med. Min hatt, den har trekanter.
Red is breathing.
Der har min hatt, og har den ei trekanter. Så den ei min hatt. Kan dere den? Ja, få høre dere. Min hatt.
There they go. Yeah, that's after they were invited to sing along. You know. So as the MC would do this, it'd be like members of the orchestra.
Ok, det nydelig, men jeg følte at
I'll stop this. There'd be
trekanter.
Orchestra that would start to kind of breathe along, and then they like switch to actually singing, and then they are all singing together, and it's really cute. And part of what is happening is that the physical synchronization of singing together is actually pretty extraordinary. And it's also funny that whether you're singing loudly or maybe even trying not to sing, there's some part of you that may be still breathing with everybody else, whether you want to or not. Thank you for asking for the cute video.
Super cool.
Yeah.